You're talking to Travis?
All right, everybody, let's get started.
Feel free to begin.
Okay, we'll get started. It's 11:30. Welcome to the WormBase workshop for the 23rd International C. elegans meeting. Thank you for everyone for attending. We hope this to be an informative and engaging workshop. As you see on the slide presented here, we have our agenda is basically we have 4 15-minute talks followed by a 30-minute open Q&A discussion. We are going to hold off on addressing questions until that 30-minute Q&A session at the end of the workshop. So, but please feel free at any point if you have a question to post your question in the Q&A box, as you should see in the Zoom interface below. I'm going to post in the chat so that everyone can see some points that I just wanted to make here, some of which I already covered. These the questions that are posted in the Q&A box will be addressed later, but will be collected and organized by WormBase staff, Daniela and Valerio. And any Q&A content, including questions we don't have time to address, will be collected in a document and posted on the WormBase blog for future perusal. The meeting is being recorded and will be available from the GSA, the conference online, the meeting program and platform until July 16th. After that date, WormBase will try to make a copy of that available on our WormBase YouTube channel. Also, I just want to point out that this workshop, as all other sessions, will abide by the GSA Code of Conduct. I posted a link to that Code of Conduct in the chat. And lastly, we have created 2 channels on the Worm21 Slack. There's WormBase and WormBase_workshop for questions pertaining particularly to this workshop. With that, I'll hand it over to Magdalena Sharowiecki for a talk about a whistle-stop tour of all data in WormBase. Take it away, Magdalena.
Thank you so much, Chris. So I am possibly one of the most recent hires of WormBase, and probably all of you who listen here, you've spent many more years than I have looking through and enjoying all of the data in WormBase. But starting to work with these guys and actually starting to wrap my head around this data has been so wonderful. We're doing a lot of talks that are focused maybe on the tools. So we thought this could be nice to just have the data in focus and try to look at all of the wonderful data that we have in WormBase. So we look at all of the papers published by C. elegans. We normalize, analyze this data and integrate it in a database. And taking a quick overview look, I realized that we're actually holding more than 22 million objects. And this is not like 22 million data points, but each object, say a gene, can contain a lot of other stuff as well. So these more than 22 million objects are in 116 data classes. So some of these data classes might be quite familiar to you, genes or strains, variants, and some of them might be a little bit more obscure. So the rest of this talk, I'm going to try to go through a fair few all of these 116 different types of data. And how has all this data come to WormBase? Well, the easy answer is that it's from all of you. The more complicated is that some of this is from analysis that we applied during the build, some is from manual curation, some is from email correspondence, author fast pass. And all of this data gets normalized, standardized, reviewed, and curated and added to the database. So I try to think of the database. Each class of these contains objects which have been carefully crafted and put there and reviewed, and an amalgamation of lots of data from lots of different sources. Breaking this down, these 22 million objects, and looking at what are these, I tried to categorize them, and I realized that the large bulk of this is genomic data, so sequences, genes, proteins. You have a fair chunk of data relating to experiments. We also have ontologies that are super important, various data classes to do with reagents. And finally, one of the smallest slivers here is papers and people, which amazingly is what generates all of this other data that we have in here. So I took the data model that we have, and what's really cool about these 116 types of data classes is that they all link together to each other. And that means that you can do really flexible queries throughout the data. So you can say which genes are mentioned in this paper or which papers mention this gene. And we have other talks like our webinar on WormMine that can teach you how to do this. But I'm going to try to focus now on just kind of showing a little bit about what is in each data point. So you can use a network like this. I think it's of a little bit like you're jumping between these different classes of data, and you can create really complex queries. Like starting with a particular phenotype, you can look for papers mentioning that phenotype, and then genes also mentioned in that paper. do those genes have any expression pattern, and are any of them expressed in the tail of a worm, for instance? And including some of these highly connected nodes into your queries will probably help you also to get better results as you're doing these kind of really complex queries. So this genomic and reagents data then, what is this? Well, starting off, we have the genome, we have the physical genome, so the actual DNA sequence, the chromosomes, the clones, the superlinks, plus a fair amount actually of genetic mapping data. Some of these data classes, they contain the very oldest data objects that we have in the database. So that's kind of cool. Then you also have some 5 million of these objects that are related to genes in some way. Genes, transcripts, CDSs, protein, and a bunch of other stuff a little bit related to that, like pseudogenes and operons. Funnily enough, the sequence is actually not a part of the protein or the CDS, but they kind of tagged onto it. So there's a link between the CDS and its sequence. UTRs, if you're looking for them, they don't really exist. They're recalculated as a subtraction between the transcript and the CDS. And you might be asking, why are there so many genes? I'm sure that's a lot more than C. elegans has, isn't it? And you're right. These are every gene that is mentioned. It's human genes we would catalog so that if you search, you can find them, and genes from a lot of other species as well. Some of these are, though, elegans, but they can be non-coding long and non-coding short RNAs. I've seen some great talks about some of these, so I know that you guys are interested. And there are several different ways that you can access and look at the data we hold about non-coding RNAs. And then continuing on the genome, there's a bunch of other things as well that are mapped to the genome or have a genomic location, but without particularly being genes. So you have transposons, you have our official features that have WBSF ID, and you can follow these, so there will be splice leader sites that we kind of quite sure about. And then we also have a bunch of non-persistent features that are called feature data, which maybe we recalculate, or they don't have that level of evidence, so we keep them as feature data rather than as named features. One of my favorite classes is genetic variation. You have more than 5 million objects here. We're getting close to 50,000 strains, 2 million plus variations, and another bunch of variation names, which live in its own little class. We have chromosomal rearrangements and very lovingly curated transgenes and constructs. And this is really a data class to watch. A lot of things are happening here right now. In the release 280, we added molecular consequences for variants calculated by the variant effect predictor, VEP. So if you're particularly looking for variants that are causing a frame shift or deleterious or pathogenic, that should be very easy for you to find now. They're in the molecular constructs hash. We're also improving our pipelines for detecting and annotating strains and variations from papers. And there will be more data for you guys to review in author first pass. We're also hoping to do better support for CRISPR-generated variations. And then, as I mentioned before, there's a bunch of species apart from C. elegans in WormBase as well. We curate gene models for all of these guys. So it's C. elegans, the N2 strain, a bunch of other Caenorhabditis, and a few important parasites as well. And then there's an additional bunch of species that we use for comparative genomics. These include C. elegans Hawaii and VC 2010 strain, which some of you also might be really interested in. For some applications, these might be the ones that you want to use rather than N2. For the 281 release, which is coming up now, we've almost doubled this amount of species with 10 new species depicting Synrhabditis genus diversity. And if that's not enough worm genomes for you, do please go over to WormBase Parasite, where you have soon close to 100 genomes of nematodes and flatworms. And far from all of these are parasites. There's a bunch of Synrhabditis and all major nematode clade data in there as well. And actually just doing a very raw lookup in the database and asking how many species are there, there's actually almost 8,000 species that have a mention. So some of these, you know, they can be the source of molecules, they can be involved in transgenes, they can be an interacting species. But if you're interested in things like microbiome or pathogen, having an entry point as species could be a really good idea, because species are part of a lot of different classes, or linked to from many different classes. And also, if you're very certain that you're only studying one particular species, you want to put in a species filter in your query, so you only get relevant data for you. Moving on to ontologies. So ontologies is a super easy way to very quickly narrow down a search. So you can say, oh, give me all of the genes, or you can say, please give me all of the genes involved with cellular developmental process. And that's where you very quickly restricted your query to the most relevant results for you. More than half of all of the classes have associated ontology terms. So very nice and quick filter. We are using a lot of ontologies that we get from other people that are internationally recognized, like the gene ontology, sequence ontology, and that contributes a fair amount of objects to our data. And we are recalculating each build and this recreating this association between these ontologies terms and the particular objects in the database. Furthermore, we have 3 ontologies that are actually ours. We've made them and we're maintaining them. And they specifically contain the kind of terms in the structured dictionary that is particular for those of you who want very fine-grained information about phenotype, development, and gross anatomy of worms. Most of this is C. elegans, but there's also other terms that are actually important for parasites and other species as well. And then we have experiments. So this is a very massive, very diverse group of data classes that really tries to show off and capture all of the important data that is generated in experiments, both reagents, methods, and results. So very rich data set, and some of the talks following on after this, they will go a little bit deeper into those. One that I particularly want to highlight is expression data, where we have 2 different tracks, if you will, for expression data that have to do with individual genes, where you can find expression patterns, expression profiles, images of particular genes being expressed. And then another track for large scale and genome-wide profiling, where there's a bunch of tools as well that you can use to analyze those. And finally, we have the papers and the people, where we're collecting papers, authors, lab, and WormBase people. So please make sure to keep us updated. And all of this data is living and changing, and we are really adapting what we are curating to your interests. So do please tell us what you want to see next. So thank you so much, everyone, and over to Chris, I believe.
Yep. Thank you, Magdalena, for a great talk. With that, we'll move over. I'm going to share my screen now. So I'm going to now shift gears a little bit and talk about transcriptional regulation and researching C. elegans transcriptional regulation at WormBase. So for starters, this is just a slide you just saw from Magdalena's talk. I, of course, want to recognize all of the staff at WormBase at our 3 centers, Caltech, OICR in Toronto, and Malcolm Trust Sanger in EBI in Hastings, England. And in particular, for this context of this talk, I would like to give special thanks to 3 WormBase alumni, Gary Williams, Xiaodong Wang, and Marianne Tooley, for all their work curating transcription factors and transcription factor data and binding sites in WormBase. So just to start, one of the classes that Magdalena referred to is the transcription factor class. It's not highly visible currently. We hope it will be in the near future. But I just want to point out that we have currently 1,156 transcription factor objects. This includes individual transcription factor genes as well as complexes. In total, there's 1,098 transcription factor genes, including, for example, one uncloned gene, and that was classified as a potential transcription factor based on gene expression phenotype. We also have 33 general transcription factor genes in there, so you should keep that in mind. It's not all just sequence-specific DNA binding factors. And that's not an exhaustive list. The full list is probably more like 80 general transcription factor genes. Now, on that note, I'm considering about 153 additional genes for consideration of addition to this class based on various TF criteria. 83 transcription factors in this class have position matrices, 174 in total, roughly 2 per transcription factor. Oftentimes, we have 1 position frequency matrix, which is based on the raw sequence alignments, and then the derived position weight matrices. These transcription factor objects include, as I said, complexes like RNA polymerase II and RNA polymerase III, which are not really transcription factors, but they're convenient to have in this class for the purposes of how we use them. There are 26 transcription factor complexes, including 5 general transcription factor complexes. These include things like homodimers, heterodimers, heterotrimers, and so forth. And for the GTF complexes, we have TF2 complexes like TF2D, we have some TF3 complexes, and I'll try to flesh that out and make those all available. In addition, we have 21 transcription factor objects that are based solely on functional enhancers, motifs, dysregulatory sites that don't have an explicit cognate transcription factor gene or protein. They're kind of orphans. We presume there's a transcription factor that binds those sites, but we don't know exactly what it is at this point. Also, there's about 30 of these transcription factors that are slated for deprecation based on review. These are either non-transcription factors, they're genes that are now dead genes or pseudogenes and so forth. I just want to point out that if you go to WormMine, which you can find under the WormBase tools menu, you can go to our lists page. And if you make sure to select the view panel on the lists tab, you can find a list of 1,075 transcription factor genes. The reason this number is not 1,098, as I explained before, is that WormMine inherently ignores pseudogenes and dead genes. So this is probably a more up-to-date and accurate list. But keep in mind that, again, there are about 33 general transcription factors in that list. I hope in the near future to create maybe 2 separate lists in WormMine that are available to the public, one for general transcription factor genes and one for sequence-specific DNA binding transcription factor encoding genes. As I alluded to, there are position matrices, both frequency and weight matrices. There's close to 6,000 position matrices in total. As I said, it's a mix of frequency and weight matrices. And as I said before, 174 of these are attributed to 83 transcription factors. These are derived from a total of 43 publications, with the vast majority coming from a large study by Gary Stormo's group, finding 5,000 motifs coming from a genome-wide analysis of conserved elements across many species and I believe Syneriditis species. The publication IDs are given there. And these position matrices are actually incorporated into JBrowse and are available for rendering on a particular track, and I'll explain that as a demonstration in a little bit. Also, Magdalena mentioned sequence feature objects. I do want to mention that these are exclusive from genes and CDSs and transcripts and these other kind of higher level or higher status classes that have their own classes. These are sequence features like binding sites and splice leaders and so forth. We have close to 1 million of those. About a third of those, 330,000 or so, are associated with the transcription factor. We have close to 2,000 directly in a curated interaction. These could be regulatory interactions showing that the cis regulator feature actually regulates gene expression, or it could be a physical protein DNA interaction. Now, in our global C. elegans GFF3 file, in particular, this is for the N2 GFF3 with that, so pay attention to bioproject numbers. If you filter column 2 in the GFF for the TF binding site region flag, you'll find that there's close to 900,000 TF binding sites. These are essentially all mod encode ChIP-seq TF binding sites. And these are all represented in JBrowse in the transcription factor binding regions track. So it's an important track I'll come back to. About 330,000 of these are instantiated in WormBase as WBSF sequence features. And they have a GFF9 column 9 format as shown here, where we name the sequence feature identifier. We provide the transcription factor ID and the name of the transcription factor at the end of the line. In addition to those, we have close to 600,000 that are from a follow-up mod encode paper. The paper IDs are given there. And these do not yet have a WBSF sequence feature, although they are represented in the GFF in the formats shown below, and are available in the JBrowse transcription factor binding regions track. This includes a total of about 104 transcription factors in the GFF that are available also in JBrowse. This is the list. I do want to point out that this list does include AMA1, which is the large subunit of RNA polymerase II. It includes RNA polymerase II complex, TATA binding protein, TBP1, and Dumpy27, which is a member, although it probably binds DNA sequence specifically, is a member of the dosage compensation complex, and may not be considered, a canonical transcription factor. So just keep that in mind. But we will share all these slides, and I would encourage you to use this slide as a reference for a query. Now, I'm going to explain how to convert this ChIP-seq data with peaks that have been called and those regions have been put into the GFF. And I want to explain how we can extract that and try to make a candidate target gene list for those data. Now, doing this, I'm putting up here 2 commands that are Unix or Linux commands. If you're not comfortable or familiar with this, please reach out to us. We can help you. It's quite trivial. Usually just open a terminal. I run this on a Mac, on my Macintosh, and that works fine. Should work on Linux. If you're using a PC or a Windows machine, you may have to just install a Unix emulator so that you can run these commands. So in this example, we're pulling from the WS280 version of the GFF3. Please pay attention to the bioproject number, PRJ-NA13758. That's the N2 canonical genome, and that has most of the annotations in the GFF. And this one is for the example of DAF16. The first command takes a few minutes to run, but once you've run that, you can run command 2 and any other subsequent commands like command 2 for different transcription factors, and they should only take a few seconds. When you do this, you'll create a TFBS coordinates.txt file that will have coordinates in the coordinate format shown below. If you have any problems trying to do this, please let us know. So we'll take these coordinates and go back to WormMine and now take advantage of the regions search. This allows you to put in a list of coordinate regions or spans and find features that overlap or reside close to these coordinate regions. So the first thing you want to do is click on the select features types at the top, which will deselect everything, and then specifically select gene. Importantly, if you're only interested in looking at coding genes near these sites, then I would recommend clicking on CDS instead of gene so that you get the CDSs and the corresponding genes there. But this choosing gene will return coding and non-coding genes. So then we would paste in our coordinates from that command I showed before. If you paste in the entire list for one transcription factor, that's likely to be thousands, and it'll take several minutes to run. It will complete, you just have to have a little patience. Once you've pasted in, you click search, and you should get back a table that looks like this, identifying the various genes that overlap the regions that you've put in. You can download this as a tab delimited file with the export features at the top. This is what it looks like if you were to open it in Excel. It lists the genes and the spans of the genes and the user input. Those are the regions that we got from the TF binding sites from the ChIP-seq data. Now you can use this, of course, to create a non-redundant list of gene IDs. You may also want to incorporate the counts of each of these genes as perhaps a metric for confidence. Importantly, when you do this with all of the regions, you're very likely to get many that don't hit, meaning those regions don't lie exactly within the extent of a gene, but in an intergenic region. In that case, you want to maybe redo the search with those regions that didn't hit and use this extend your regions at both sides feature. You can type in a value like 1,000 base pairs or use the logarithmic slide scale to slide to the size that you want to include, and then click search, and then you will get genes back now that include genes within 1,000 base pairs on either side of the region that you put in. Some caveats to this approach are that this method does not discriminate between upstream and downstream binding site locations. Unless you choose the CDS option, if you just choose gene, it will include both coding and non-coding genes. Some regions may require different length extensions to find the nearest gene. So you can use an iterative approach so that you don't get you don't cast too wide of a net initially, and you could keep expanding your region, but that can become quite laborious, just so you're aware. And as usual, of course, unambiguously assigning an intergenic region to a gene solely based on location is still quite a challenge. Now I want to shift gears and talk about how we can use JBrowse to find potential TF regulators and targets. I'm going to start with the ATFS1 gene, which is a transcription factor that's critical to the mitochondrial unfolded protein response. If we go to that page and say, click on the genomic position, we can open that JBrowse centered at that gene. I want to point out that in this left panel, the data for transcription factors will largely be in this mod encode data section. And in particular, look at these GFP chip and GFP chip peak recall tracks. Those are the tracks you really want to look at the ChIP-seq data and the peaks that have been called for them. There's a section called Transcription and Chromatin, which is a little bit misleading. I think that there's some tracks in there that are labeled transcription factor. I think that was a large a generalization, general category of how the data was submitted. Most of the things under that category are things like cohesin and condensant complex members and so forth, and chromatin factors. So really for the sequence specific DNA binding factor, transcription factors, I would look at the GFP chip tracks. Also, here's the, if you come down further, there's a sequence features section, and in that is a binding sites and regions. Importantly is this transcription factor binding regions. All of the features that are TF binding sites in the GFF that I mentioned will be found in this track. So if you turn that track on, we can see in the region of the ATFS1 gene, there's many different binding sites. Some of these are repeat elements, some of these are polymerase II, so keep that in mind. But if you want to get the full list here of all the potential transcription factors that bind in this region, you can use the highlight tool, highlight the region that spans on either side of the gene, and then click on the track and click on Save Track Data. This will default to the highlighted region and to the GFF3 format. Save this, and you should get a GFF with the column 9 format looking like this that I described earlier. And this should list all of the transcription factors that are that are binding to that region. Additionally, I want to point out that there is a Add Motif search track. If you click on the File option, This gives you a list of preset motifs or position weight matrices or frequency matrices that have been pre-published and have been made available and incorporated into JBrowse. You can provide a custom input for PFMs or PWMs, and then, excuse me. In this case, I'm going to choose as an example the Fa4 motif from AO 2004. And importantly, you need to put in a minimum score to match. So on a scale of 1 to 100, I'll put 90 to only return high scoring matches. I'll click search, and it may be hard to see, but right here are 2 predicted Fa4 binding sites based on the motif that we have in the position weight matrix. And sure enough, if we pull up the Fa4 SHIP-seq peak tracks, we can see 2 distinct clear peaks at those predicted binding sites in both L1 and L4 life stages from the mod encode data. So that's a nice corroboration. Importantly, it's important with all of this data to make sure that you corroborate effectively with the various types of data and how it comes in. Doing large scale analysis can be nebulous and tricky. You have to take into consideration many caveats. Finally, I'd just like to finish by talking about the Alliance of Genome Resources. You can find now on our Alliance gene pages for C. elegans genes. I'll use ATFS1 as an example. You can come to the ATFS1 page of the Alliance, go to the molecular interaction section, and then in the interactor molecule type, you can filter just for DNA targets. And so this will return a list of 793 putative target genes for the ATFS1 transcription factor. Alternatively, you can turn off that filter and say you want to find interactions for which ATFS1 plays the role of the DNA interactor, and then you will return a list of 19 putative transcription factor regulators in this list. Importantly, no single method or curation approach captures all the TF regulatory or target data. There is an overlap, but largely data will come from one type or the other. It's best to look at all types for evidence and corroboration. And with that, I thank you for your attention and remind you to please stay in touch by emailing us at help@wormbase.org or curation@wormbase.org as needed. Thank you, and I'll turn over the session to Ranjana. So Ranjana Kishore is going to talk about the Alliance of Genome Resources, in particular, using it for disease models.
Hello, everyone. I'm Ranjana Kishore, and I'll be telling you about the Alliance of Genome Resources and the great comparative views we have there. And I'll tell you about this in the context of disease research data at the Alliance. So my presentation mode, Chris?
You have to click present at the top right.
Okay, view.
Or you can go to view.
Yeah. Okay, so the Alliance of Genome Resources is a consortium made up of the member databases, which are the major model organism databases that this audience needs no introduction to, as well as the Gene Ontology Consortium, which annotates gene functional data from many organisms, including the major species of the Alliance. So the guiding vision of the Alliance of Genome Resources that was first released in October 2017 was to provide a single comprehensive platform, highly integrated, to provide comparative views of data from the major model organisms. This includes sort of providing a single point of access to multiple types of data from all these species, providing harmonized and unified views of this comparative data with sort of interactive tables and lists. We provide links to source databases and external references where it's relevant. Access to the data at the Alliance is through data downloads and application programming interfaces, commonly known as APIs. The data is refreshed through quarterly releases, And even as I speak, new data types and data displays are being worked on. So we like to think of the Alliance as a one-stop shop for genomic and genetic data, and some of the data we have relates to these, what we call genetic entities, such as genes, alleles, variants, transgenic alleles, strains, and genotypes, and then sort of the data that projects off of these, like orthology, functional data, which are the gene ontology annotations, phenotypes, disease associations, expression data, molecular and genetic interactions, and we have other tools like the Alliance Mine. So you might have heard of like mine, various mine instances. The Alliance has its own instance known as Alliance Mine. So today I'll be focusing on mainly genes and disease associations. And then, of course, the underlying orthology that sort of is used to provide all these comparative views. So this is the landing page of the Alliance. And please pay attention to the version of the Alliance. If you use the Alliance and you need to cite it, it is always good to mention the version that you used. So what I'd like to do in this talk is to do two things. First is to show you how the Alliance can be used to study human diseases and how you can access the human disease research data. And by doing this, I also hope to illustrate some of the principles that the Alliance uses to sort of present the data and how the data is organized. So let's take a real world example. So you're a worm researcher and you're interested in using the worm to study Parkinson's disease and the genes involved in Parkinson's disease with a special focus on LRRK2. And LRRK2 recently has gotten a lot of sort of attention. It's a little bit still of a mystery. By the way, LRRK2 is a leucine repeat kinase 2. Leucine-rich repeat kinase 2. It's still a bit of a mystery. It's expressed in like sort of very widely, yet its mechanistic action is still not completely elucidated. And it's sort of like a hot target for a lot of therapeutics. So how can the Alliance help you find Parkinson's data from model organisms? So that's the two questions I'll be addressing. So sort of the genes involved in Parkinson's disease, and then how to get at Parkinson's data within the Alliance. So this is the landing page for the Alliance. And so you start with the gene of interest, which is the human gene LRRK2. So the powerful top level search offers a drop-down menu. You choose the human gene and you arrive at what we call a gene report page for the human LRRK2 gene. So these report pages in the Alliance, whether they're gene pages or disease pages or sort of information hubs that connect a lot of the related data. And all of this data is sort of brought together by the great comparative genomics underlying all of this, that is the orthology. So one of the early goals of the Alliance was to sort of come up or develop a set of orthologs, common orthologs across the species, the human and major model organisms, and to use this common set to provide comparative views of all the related data. So on the human LRRK2 gene page, since we are C. elegans researchers, we are interested in the C. elegans gene, and that you can find in the orthology section. So following the link from the LRRK1 C. elegans gene, one arrives at the C. elegans LRRK1 gene page. And on the C. elegans LRRK1 gene page is a lot of information about the gene itself. As I told you, there are cross-references to external resources. There's an automated gene summary, which It gives you like a bird's eye view of functional data. And within this automated gene summary is disease relevant information as well. And it already says that it's used to study Parkinson's disease. So you already have some idea that there are probably models of Parkinson's in C. elegans. So in the orthology section, now sort of zooming in into the orthology section, there are many interactive features. For example, you can open these additional filters to constrain the data in additional ways. For example, there's a menu that opens. You can click on best score, best reverse score. You can constrain the data by species. You can constrain the data by your favorite method. And you can also follow this link here to get a phylogenetic tree view of LARC1 at the Panther database using the Panther classification system, and you can explore that data here. So coming back to the orthology view on the C. elegans LARC1 page, just a little bit of information on how orthology is computed at the Alliance. You can use this little tooltip. Clicking on it will open a box with a lot of information of how orthology is computed at the Alliance. And to make a long story short, we actually use Diopt, which is a tool that sort of integrates all these methods, these great orthology calling methods. And the Diopt approach actually offers flexibility in the choice of algorithms and in the level of stringency applied, though the default view is always the most stringent view. And for the orthology table here, the user can sort of specify these different parameters. So that was a little bit of tangential information about how orthology is computed. So apart from orthologs for LARC1, the Alliance also has on the LARC1 gene page for C. elegans, it has disease associations. And just a note to say that disease associations at the Alliance are represented also by an ontology, just like other types of data. And the ontology we use is known as the disease ontology, and we use DOE terms to describe diseases. The disease ontology is developed by the Disease Ontology Project at the University of Maryland. So in the disease association section, we also provide graphical views, what are known as ribbons. And the ribbon was first developed by MGD, later adopted by GO, and now by the Alliance to sort of graphically represent different types of data. It's a sort of a matrix-like representation. The subsections sort of group related diseases. The shaded cells indicate the presence of disease annotations, and the darker hues indicate more data. So this is sort of an interactive system where you can click on a cell and then bring up annotations for that species, or you can also leave it on the default view and then filter by these filters on the table. And again, we have a comparative view across species. Remember, we're still on the LARC1 C. elegans page, and yet we have this great comparative view across species. So just a little bit of the interactive features. You can further constrain results by using the species filter. And here I've chosen the Homo sapien, and therefore you get all the annotations associated with the LARC2 gene in human. There's an option to include negative annotations. By negative annotation, we mean that they were expecting to see probably a phenotype associated with Parkinson's, and the authors did not see this, so they used the association type is not implicated in, and then you can go to the reference to see more of that data. So now that we covered the disease associations, and as I said, the example was that as a C. elegans researcher, you were interested in the LARC1 gene, but you're also interested in other genes that are involved in Parkinson's. So how do you do that? So the easiest way is, again, to go to the top level search, the global level search at the Alliance, start typing in Parkin, it auto-completes for you. Using the disease ontologies, using the underlying disease ontology, and then you arrive at the Parkinson's disease report page. So again, as I said, the disease report page serves as an information hub, and it's sort of consolidating all this data related to genes, alleles, and models. By models, we mean at the Alliance, these are strains and genotypes. So again, you can use the filters at the tables to constrain your data. So since we are interested in C. elegans, you can choose the species C. elegans and get all the genes that have been associated with Parkinson's disease. You can filter by association type. So this association type describes the relationship between a gene and the disease. So here I want to get all the genes implicated via orthology. And so I can click on that and get all the genes, that have been associated to Parkinson's via orthology. And it's based on, like, what gene in what species, and then it's associated with the relevant reference. So there's also a download data option. So you can download all the data in any table by using this, and it downloads into a TSV formatted file. You can sort by disease subtypes, where the disease and its child terms appear in a table. So it's sort of educative in itself, telling you that Parkinson's disease is further sort of divided into all these subtypes. So on the disease report page, you can also explore, it's not just genes, so I just wanted to describe the interactive features using genes. You can also explore associated alleles and associated models, which are strains and genotypes, and then follow the links and explore the data from there. So disease data can also be accessed sort of in bulk via downloads and APIs, as I mentioned before. So that can be accessed by this menu, data and tools. So when you open that, you can follow the link and go to the downloads page. And the disease data for each of the species of the Alliance is available in both JSON and TSV formats. You can also explore the different endpoints we have via the Alliance APIs. So you can open the disease, look at all the endpoints, and sort of, get your hands wet and just sort of like play around with the endpoints to sort of get the bulk data. So what are the future plans for Alliance? So we will be harmonizing and including even more data types, example pathways, inducers and modifiers of disease, richer variant data. We hope to build model organism portal pages, disease portal pages, where we will be building sort of synthesized views of the data. For example, maybe you can query the Alliance for questions like, give me all the genes involved in Parkinson's but are expressed in the brain in the species. So sort of a more synthesized view of the data. In addition to that, we hope to add additional species such as Pombi and Xenopus. In addition to all the interactive features, we also have tutorials, user documentation, and FAQs. You can get at those by opening the help menu, and here are all the Twitter, Facebook, and email information. You can email us at help@alliancegenome.org. You can join sort of a very active Alliance user community. You can access official announcements, ask questions, and participate in discussions along with other members of the Alliance community. So I'd like to especially thank the Alliance Disease Working Group of the Alliance and colleagues there, and of course all my great colleagues at the Alliance, and of course all my colleagues at WormBase, and especially the PIs that have offered us great support to do this work. Thank you, and back to Chris.
Okay, great. Thank you, Ranjana. With that, we'll move on to Kimberly's talk. Kimberly, just so you all know, we had a change of speaker. Instead of Daniela Ricciuti, we'll have Kimberly Van Auken talk about community curation and ways that the WormBase community can contribute to WormBase data. I can see your slide, Kimberly. You can go ahead.
Okay, great. Thanks, Chris and Ranjana. So for our last talk of the WormBase workshop today, what we'd like to tell you about is how you can contribute to WormBase and the various tools and methods that we have for community curation. So community curation is really important to projects like WormBase for several reasons. First off, you as authors are really the subject matter experts. So although as curators, we try to cover as broad a range of information as possible, we know that our authors who are working really hard in the lab to study their particular areas are really the experts on the biological data. Secondly, as Magda showed you in the first talk, behind all of the great WormBase web pages is a structured data model for each of the data types. And so with our community curation pipelines, to the extent that we can get our authors to fill in structured forms, that makes it much easier for us to integrate the data into WormBase. And lastly, unfortunately, we do have a bit of a curation backlog. So community curation is a great way to help us move more of our papers into the done pile. So there are two main ways right now that people can contribute to WormBase, and I'll talk about each of these in turn. So basically they break down into post-publication and pre-publication. So post-publication means that your paper has been reviewed, accepted, and pretty much it's out there, say, in PubMed or on the journal website. And the two main ways you can do this that I'll talk about in a bit more detail is through our author first pass pipeline and through some data type specific curation forms that we have. In contrast, the micropublication biology journal is developing ways that authors can contribute curation pre-publication. And so I'll talk about that at the very end. So let me start with author first pass. So author first pass basically is just a way for you to tell us what's in your paper. And the way this works is once your paper has been published, we have a dedicated team member at WormBase who goes out and gets the PDF of your paper. We convert that to text, and then we use as much natural language processing as we can to identify the types of data that are in the paper and also the authors to contact. So in terms of what we try to identify, we break this down into two things, basically entities, which are correspond to a lot of the classes that Magda mentioned in her talk. So these are things like genes and alleles, strains, transgenes, species, things like that. And also more generally, what we call at WormBase data types that roughly correspond to the kinds of experiments that people do. So there might be an expression pattern or maybe mutant phenotypes, physical or genetic interactions, those kinds of things. And then lastly, we look for the corresponding authors and we match them with an email. And then once your paper has been processed, we'll email a form link to the authors that will look like this. So in your inbox, you'll see an email from Outreach at WormBase with the subject, help WormBase curate your paper. And so basically in this email, there is a link here, and that will take you to a form specifically for your paper. And I'll show you what that looks like and what you'll need to do. We hope that this process will take 10 minutes or so. It really depends a bit upon the complexity of data that's in your paper. And lastly, it's important to know that if you aren't able for any reason to fill out the form, it's perfectly fine to forward your email to another co-author. As long as they have the link, they can go ahead and fill out the form. So when you click on the link, this is where you arrive. This is the author first pass landing page. You'll see that it will list the paper. It will automatically list a person ID for the corresponding author. If you're not that person, but you're filling out the form, you can just change that. And so we always know who exactly has filled out the form in case we need to get in touch. So the author first pass form is generally organized according to data types, and we've roughly tried to follow the organization of data in WormBase as well. So this just shows you an example of what we're asking authors to do. So like I said, we try to use our text mining applications to identify as many things as possible up front and then just ask authors to verify the lists. So this is an example of all of the genes that we've identified in the paper. Particularly what we're looking for with this are genes and variations and strains and transgenes that have really been used for experiments in the paper. So for example, things that might be used as tissue-specific markers or genetic markers aren't necessarily the things that we want to associate with papers in WormBase. But as an author, you can go through and check the list. And if there's anything that our text mining identified that isn't a real positive, you can remove it from the list. Likewise, if we've missed things, you can add them. And as much as possible, we try to use autocomplete from carefully curated WormBase lists to make it easier for people to accurately enter new entities. The second part of author first pass is what we refer to as data type identification. And like I said, this really roughly corresponds to the kinds of experiments that you do. And so in this case, if we think that a particular data type is in the paper, it will be checked. If we're wrong, you can uncheck it, and that gives us really valuable feedback to improve our methods. If we miss something, you can check it. And again, that's great feedback for us. In many cases, if you want, you can add free text details. Hopefully in the future, what we'd like to do is develop more and more structured curation forms that if authors want to add details, they can do that in a way that really seamlessly integrates that into WormBase. Lastly, when you're all done, you'll get to this last widget here and you'll submit the data. And WormBase curators will go over that as soon as possible and get that data into WormBase. So as I mentioned, another way to submit data post-publication is to submit specific types of data for our, that we have dedicated curation forms for. So you can do this, one way you can do this is from the WormBase homepage. You can click on the submit data link. And that will take you to this page where you'll see we have dedicated forms for specific kinds of data. I'm going to show an example using our phenotype data submission form. So this is what this form looks like. This is just really part of this form, but you can see again, we look for just your contact information. So if we have any questions, we can get in touch. And you can give us specific details about, say, the genetic perturbations, the phenotypes, using the phenotype ontology that Magda mentioned. And then if there's additional information you'd like to give us, there's an optional section to do that. So the phenotype curation is another pipeline where we email authors every few months to ask for community annotation from specific papers. One key difference between this and the author first pass is that for phenotypes, we will ask for curation of both old and new papers. And like I said, this will really help with our curation backlog. So we've recently implemented some new features into WormBase to recognize our community curators. We really appreciate the effort that you put into helping us get information into WormBase. On the homepage, and this has been around for a while, we show the top activity for our phenotype curators, but then also recently on the WormBase person and reference pages, we've added a community curation section where we thank authors for contributing to our community curation pipelines. And if a publication has been community curated, we'll indicate that on the bibliography pages. Okay, so lastly, I want to move and talk a little bit about pre-publication curation. So this is work that's being developed by the micropublication biology term. So this is a novel publication platform, although we're starting to think that this is actually becoming a well-established publication platform. And as Paul mentioned in his plenary talk yesterday, 5% of the C. elegans papers that were published were from micropublication biology. So basically, again, this micropublication biology was set up generally to capture single experimental results. And it's really a great way to capture things that perhaps might have otherwise gone unpublished. We know that not all of the information that's generated in the lab always fits neatly into a larger paper. So this is a great way to get that information out. All the micropublication biology articles, they're peer reviewed and they are now indexed in PubMed. It's nonprofit and open access, and it really facilitates, again, efficient incorporation of new findings and new data into scientific databases. But just like WormBase in general, micropublication biology also need authors' help to curate. And so they are developing some easy community curation tools. And so they've shared the screenshot of the kind of tool that they are developing. So the idea is that once your paper has been reviewed and accepted in micropublication biology, they will then be asking authors to help get the information into WormBase in a structured way using this curation tool that they're developing. And so like the author first pass, we often use what we would call entity recognition to find sentences within the text of your publication and recognize the entities that are in those sentences, the kind of data that this sentence is describing, and then put this into a very nice, easy to use tool for authors to then verify and submit your data. So in this example, say for an expression pattern of UNC59, you would select the gene. You would select the anatomy terms and life stages for that expression. So this is a 2-cell embryo, corresponds to A, B, and P1. And then where is this localized? It's localized in the cleavage furrow. So this again is a really nice way. Hopefully we're making it easy for authors to tell us the data that's in your paper. We can get that in a structured and quick way as possible. So just to summarize, so we have two main types of post-publication community curation pipelines: author first pass and data type curation. And the micropublication biology is a new way for us to get data into WormBase pre-publication. So while I was preparing this talk, I went looking to see what participation rates are at various mods. So WormBase isn't the only model organism database that has a community curation pipeline. Other great projects like TER and PalmBase and FlyBase also ask their authors, but as you can see at WormBase, we're lagging a little bit in our response rate. And it would be awesome if in the next international meeting in 2 years' time, we're up there with all of these other projects. And so finally, again, just an acknowledgement, WormBase is very much a team effort. And so we really appreciate all of the efforts of everyone in our group. And with that, I will stop and we'll be happy to take any questions.
Okay, great. Thank you, Kimberly. So we're we're now going to enter our Q&A session. We have about 25 minutes to do Q&A. Daniela Ricciuti, I think, is going to field the questions. They've been cataloging the questions that you've been putting into the Q&A box. And we're going to try to divvy up the time equally across the topics and talks and general questions. So Daniela, I'll hand it over to you and we can try to address the questions. If all the panelists could turn their video on and be prepared to answer questions, that would be great.
Thank you very much, Chris. Thank you to our speakers. We will address a couple of questions that came in for each panelist, and then we'll move to the general questions, as Chris said. Please continue to type in questions in the Q&A window. The first question we got is from Lucas Morgan. Lucas is saying that he often cannot find correct promoter information on WormBase via the transcript page. He's asking if he's looking in the wrong spot or if this is not currently covered on WormBase. If not, if it's possible to extract this from published papers. And I direct this question to Paul Davis, our sequence curator at Hinxton. Paul?
Yeah, so I was having a quick think about this. But basically, we did a pilot study to sort of do manual curation from publications and identified that the way people sort of talk about promoters within publications, it's quite variable. So we did do some curation, but it's not a huge number of genes are covered in that curation effort from a few years ago. So if there's specific sets of promoters that are available and out there, maybe this is something that we can sort of revisit and sort of come back to. Obviously, there's lots of data on JBrowse that gives you kind of like hints as to sort of where promoter regions might be, but the actual sort of curation of features dedicated to promoters, it isn't very sort of rich at the moment. As I say, yeah, if you've got specific things that you'd like to point us to, then we're more than happy to sort of revisit that.
Thank you, Paul. And
yeah, I'd just like to add to that. This is a common question, and as Paul says, it's tricky, and the gene models change frequently. We have some promoters from the promoter-owned project in the promoters track, and they're instantiated as WBSF sequence features. I'm going to post in the chat. Hopefully, I can get it to everybody, analysts and attendees. There's a tool called RSAT. Just put it in the chat. Hopefully, you can see it and link to it. They have a sequence tool called Retrieve Sequence. The cool thing about that is you can go in and put in a list of genes, and it will return upstream sequence, excluding upstream open reading frames. So if you just want intergenic sequence, that's one tool that you can use. Thanks.
thank you, Chris. So the second question is from Wouter van der Berg. Wouter says that in Magda's presentation, she mentioned that UTRs don't exist in WormBase, and these are recalculated for every release. Wouter is wondering how this determination is done and what is based on. Magda?
Paul, do you want to take that one, sir?
Oh, yeah, I can do. So we have a pipeline that, curators curate the actual coding sequences, so the CDSs, the genes that sort of we come across through sort of general curation or from publications. But we have a pipeline that generates like predicted transcripts from using the CDS as like a seed. So it will take into consideration lots of sequence support, so ESTs, mRNAs, things like that. Sequence features, so SL1 sites, SL2s, things, and it will generate lots of different transcripts for a given gene. And then what we do is we subtract the actual coding sequence from that transcript and include those in our GFF and on our genome browsers as UTRs. We also have UTR-owned data from Marco Mangone's lab. So I think we've got recently it was updated to the version 2 data. So there's kind of static sets of UTRs as well as like the predicted or evolving ones that we predict during our production cycle.
Thank you very much, Paul. I hope that, that answered your question. A couple of questions from John Murray to Chris. Given that a much larger proportion of transcription factors than 83 in WormBase have had binding inferred by PBM or cross homology, it would be really helpful to be able to analyze this broader set in WormBase JBrowse. And if there are plans to incorporate the transcription factor binding site from the modern consortium, in addition to the modern code data into WormBase.
Yeah, those are both great questions. We don't have a current plan of incorporating the modern data, but we certainly want to incorporate it. As I mentioned, the people that largely did a lot of this transcription factor work have now left. Gary Williams, who did a lot of the work incorporating the sequence features from the ChIP-seq data from modding code, retired in November. As soon as we can free up some more time for curators to dedicate themselves to that type of effort, we would certainly like to do that. Same with the position weight matrices. We would love to pull those in. We will take a look at them and see to what extent we can automate the incorporation of all of that data, because I agree it would be great to have that fully incorporated now that there's such comprehensive analyses out there.
Thank you, Chris. A couple of questions for Ranjana. Ranjana, was the question from Jean about LRK1 addressed during in the chat?
Yeah, I'll address it now. So I think Jen is referring to the Parkinson's disease page, where all the genes are listed. And the reason why LRK1 is not, the C. elegans LRK1 is not at the top is because there the genes are sort of listed in alphabetical order. So you get like a lot of genes listed before you can see LRK1. And also there are only so many rows that show as the default, but you can increase the number of rows and then, all the lists will, the entire list will show. But on the human LRK2 page, because the orthology stringency is set to the most stringent, you can see that C. elegans LRK1 comes up as the best ortholog.
And I'll just add that you can use the filter options at the column headers to filter. So if you want to look for LRK1 in the data, you can go to the gene column header, use the filter, type in LRK1, and you should get all the rows that pertain to that for that disease.
Thank you, Ranjan and Chris. Another question is from Michaela. Michaela asks if we plan to include Arabidopsis as a model organism. Ranjana answered in the chat, as far as we know, there are currently no plans, but as Alliance expands, that might certainly be a possibility. Moving on to the AFP part, Brian asks if the author first pass goes to all the authors of a publication or only to the corresponding author. What we do is that we contact the first author that is associated, that has a WB person ID, a WormBase person ID in WormBase. And we manually curate person objects to make sure we have up-to-date email addresses. What you can do is that you can forward the link to any collaborator, a student, so they can fill out the form as well. You can change an author via the change author feature so that we know exactly who has filled out the form. Also, Anita asks how late is too late to do author curation for an article, and it's never too late. So Anita, feel free to get in touch with us. If you found an old email, please go to the form. If you're having issues, please get in touch with us at help@wormbase.org. I didn't see any other AFP related questions, so we can move to more general questions. Marty was asking if we are curating papers in bioRxiv or other preprints. And the short answer is we don't, but what happens is that sometimes we get a head start from authors, like the SenGen group contacted us saying that they had a preprint and their article was under review. So WormBase curator can start looking at the data and communicating with the group and see how to best incorporate the data upon acceptance. Kyriaki was asking if we have, this is a general interest for everyone if we have tutorial videos and on how to use some of the WormBase main features. Sorry, please check the Q&A. We have posted a couple of links with YouTube tutorials and also videos and slides of our WormBase webinar series that happened during 2020 and 2021. And now I'm gonna read the questions that came in while I was moderating. So Avery Davis asks, given a list of genes, what are some of the best or your favorite ways to use WormBase to figure out how or if they are connected? For example, functionality by ontology, co-regulated by location, or however.
I can try to field this one. So it's a very broad task that you're asking for, but we have a number of different tools that can get you various information with the gene list. It's really useful to use SimpleMind. You can put in a list of genes and get back a list of interacting genes. I think we include some orthology in there, but you can get a list back of interacting genes and the manner in which it's interacting with those other genes, if it's a physical interaction, a genetic interaction, or regulatory. Also, I would encourage you to use the gene set enrichment analysis tools that we have so that you can see if there's an enrichment for a certain type of anatomy term in which these genes are expressed, or a particular phenotype that tends to recur more frequently than expected by random chance in that set, or any gene ontology terms that come up at a higher rate than expected by chance. So I would encourage you to use those tools. WormMind is also a useful tool. You can input a list of genes, save it, name it, and then you can do a number of different queries constraining on that list to see if you can find similarities.
Great, thank you, Chris.
Hope that answered it. Yeah.
Next question is from Brian. If you can help curate papers that you are not authors on, and the answer is yes, definitely. You can fill out data submission forms on the website directory. So you go on directory tools and click on submit data. There might be also like a direct submit data button on the homepage. You can also email help@wormbase.org and we can give you further instructions. The next question is from an anonymous user. This is more of a general question. Is there any way by which intergenic variants on specific genes and intergenic variants can be extracted from WormBase? Magda, do you want to take this one?
Yeah, sure. The most straightforward way is to go to the FTP and download the annotation GFF file. It has the genomic location both of genes and variants. So then you can, very much decide exactly which part that you want to look at. I think potentially also if you go to the gene page, you probably get variants that are overlapping with the full gene for the intragenic variants. And finally, you also have the option of just going to JBrowse and look up your particular gene and then look at the region of interest. And then from JBrowse, then you can download all of the features that you have in your view. So that's a few different ways you can do that.
Thank you, Magda. Another anonymous question, are there any resources for phenotypic data associated with specific genotype combinations? Chris?
Specific genotype combinations. I'm not sure I understand the question. Are we looking at genetic interactions or are we looking at something more specific?
We can wait for further.
Maybe, yeah, maybe we can come back to that. If you don't mind clarifying, whoever wrote that.
Yes, so
yeah,
whoever posted the
it.
question, please put more information. The next question is for Dana. Is there a way to easily look at alignment of certain genes across all nematode data? Search for a certain gene and be able to look at the DNA protein sequence alignment in all nematode species you have annotations to. She says that this exists in the yeast community. It's very useful, but she couldn't find it on the WormBase website. Someone at Hingston. I don't think this is available, but.
Yeah, so a long time ago, we used to provide, I think, cactus alignments between some of the nematodes, but that's not that's something that we don't offer at the moment. Yeah, it's a good question and a good suggestion, and obviously something we can look into. But yeah, it's not something we currently have.
Something like that might turn up at the Alliance webpage as well. I've heard rumors about it.
Thank you. Another question for you, Chris, an anonymous user. Can we find microRNA data on WormBase, like the putative binding site and the potential microRNA binding to a particular gene like we have in target scan?
Yeah, I want to share my screen for this one. So if you open JBrowse, and I believe these should be available in the GFF as well. It's for this particular purpose, it's a poorly named track, but if you go to binding sequence features, binding sites and regions, there's a track called binding sites predicted. There's also binding sites curated, which is much more sparsely populated, but you can find predicted microRNA target sites listed. For example, this gene is T20F7.1, and it looks like the 3' UTR has a bunch of predicted sites. It's also overlapping the 3' UTR base CS9. But if you put up that binding sites predicted, you can see various different types of predicted binding regions throughout the genome. I think that's the only place we have predicted microRNA targets, but I think that should also be able to be derived from the GFF programmatically if needed.
Thank you, Chris. We have another anonymous question. When will the discourse-based Alliance Community Forum replace the WormBase forum, or is the Google group the definitive version? I don't know if Todd Harris is on the call. I haven't seen him.
No, I don't see him. My understanding is that eventually the discourse will take over and be the mechanism for chatting or communicating not only within WormBase, but also with all the Alliance organization. So I think that's being adopted as the, at least currently, for the communication forum for WormBase and the Alliance at large, so that all these communities can work together, as C. elegans researchers or nematode worm researchers, but also in conjunction with the other model organism research communities.
Yeah, I think that's coming in the near future.
Thank you. Victoria is asking, where will the slides from today's talk be posted? So Victoria, the slides will be, and the recording will probably be available via the conference website. We will also post the slides on the blog alongside the Q&A doc that we are compiling. So in case we don't manage to get to all questions, you still have a chance to take a look at the document. So please keep an eye on the WormBase homepage and new blog posts. Moving on, Ashku is asking if we have tutorials to use Alliance Mine.
We currently, oh, go ahead.
I was just going to say the Alliance Mine homepage has, on the right-hand side, has a link to video tutorials, and that link actually takes you to the example for SGD. So you can play around with that and just learn to generally follow sort of mine principles and how to query mine instances. Chris, you can add to that.
Yeah, I was going to say the same thing. Also, there's if you go to WormMine, there's some tutorial information there that can and some wiki page documentation that should apply generally to the function of the InterMine interface in general, and therefore Alliance Mine as well. But that documentation is actively being developed.
Thank you. Another mining question: to use WormMine's query builder for setting constraints, is there a way to see possible values? For example, what life stages or what expression patterns primary identifier are valid?
Yeah, for small control vocabularies and small lists, that should be built in as a drop-down menu to select various options. But for larger categories like life stage or any other terms that have a much larger vocabulary, I guess what I would suggest is before doing your constraint query, do a query on that other class like life stage to see what's there and filter on the name of the class and just peruse that to get more familiar with that class and what's available. And we don't unfortunately have like an autocomplete or auto suggest feature for constraints in InterMine. I don't know if that will be made available in InterMine in the future. But for now, I would suggest if it's a large list of possibilities, query that class separately and peruse the data so that you know what values are valid.
Thank you, Chris. Elsa is asking how to connect the genome with the connectome databases in the same idea that has been implemented by the WormWeb developed by Nikhil Batla. We can easily visualize the elegans neural network and search for which neurons express which genes, but a tool that will be updated by the community like it is done here in WormBase. I don't know if Raymond Lee is panelist.
He's not a panelist, but we only have a few minutes, so I don't think we can.
Right.
If Raymond can put an answer in the chat, maybe to everybody, maybe we can see it.
Yeah, maybe we take the very last question. There are 2 to go. Let's see if we can make it. Is there a tool to know whether a point mutation in the disease-associated gene, which causes a range of phenotypes in the nematode, is conserved in other animal models? Ranjana?
I think the Alliance is the best site to do this. Just recently, a lot of variant data was added, so you could go to follow the allele links to the variant data, and there it should be possible to sort of link to the point mutations, the type of point mutations, and then sort of navigate to the phenotypes. And if it isn't there, it's coming very soon, and the interlinks between these data. But Chris, do you know about the point mutation to phenotype links?
I mean, that should be available via most of the just general phenotype display. But I imagine the question is really how do we compare this mutant across species? It's actually a high priority item for the Alliance to be able to, I think Ranjan already said that, to be able to align all of the Alliance genomes and see what synonymous mutations may exist already or what might be candidates for CRISPR targeting. I do want to point out we have probably less than a minute before this will automatically shut off. So before that happens, I just want to thank all the presenters and everyone for attending. We will try to gather all of the questions that have been asked that we haven't been able to get to, put them in a document and make them available via the blog. If you can't find it on the blog or you missed the blog post, please email us at help@wormbase.org and we'll try to guide you and direct you to the appropriate documentation. With that, I think we're just about to end. So thank you and enjoy the rest of the conference.